Okay, so a bit of a highlight reel for chapter 26. So for chapter 26, this is one where we're not introducing a new system. It's really a synthesis chapter where we're bringing a lot of information about how we maintain homeostasis for fluid concentration, solute concentration, things like that we've seen in other systems, respiratory system, urinary system, etc., and how they work integratively with each other to help maintain homeostasis in this way. So there is some new information here, but for the most part it's review, which is always a nice thing. Okay, so the major topics, things you want to focus on here is, number one, the fact that you want to know where we're keeping all of these fluids. So you want to know the difference between, so for instance, the intracellular fluid and the extracellular fluid, right? Intra means within, extra is outside of it. So we can see that those are not exactly the same. There's more intracellularly compared to extracellularly, right? When we look at the extracellular fluid, most of that is the interstitial fluid, the fluid that exists in the space in between the cells, okay? And then about 20% of that is the plasma. So you want to know your compartments. So we talk a bit about that. You also want to know some standard definitions in terms of what makes up an electrolyte versus a non-electrolyte. And this figure, which I take from a different textbook, I think does a good job of showing you these different compartments. The first two of which are extracellular, the plasma and the interstitial fluid. And then the last one is intracellular. In this case, the example of within a skeletal muscle cell. And these are showing you the relative distribution of our positively charged cations and our negatively charged anions. And you can see that between these two extracellular compartments that are separated by the capillary wall, they're very similar in terms of their concentration of these electrolytes. But we compare that with inside of the cell, right, where we have the plasma membrane separating inside and outside the cell, we can see that the inside of the cell is very, very different compared to outside of the cell. Remember, we have different types of transport proteins, etc., that lipid bilayer, ways that we can modify and control the concentrations of these different ions inside and outside. And you can see here in terms of the size of these bars, the relative amount of these different ions. And that also goes to, probably their importance as well. So the bigger the bars that we see here, sodium, potassium, things like that, those are some of the things that we want to focus on in terms of how we control them and what they do in the body. Now, before we get to solutes, we also look at water balance. So you should be able to describe the major ways that we can lose water and kind of not necessarily the actual amounts, but which one contributes most, etc. Now, in terms of controlling and maintaining homeostasis for water as well as the solutes, you should know where in the body we're controlling it. So for instance, we're talking hypothalamus for water balance in the body, and then how we control it as well. And usually that's going to involve different hormones. So in the case of, for instance, water balance, we have ADH or antidiuretic hormone. So you should know its role in terms of helping with water balance. There are some terms that you're going to see as well that will be important. Edema is one of them, to be sure, wherein this balance between the fluids found in the extracellular and the intracellular environment are off a bit. But then we move from water to electrolytes. So again, important charged particles that are maintained at certain concentrations either inside or outside the cell. So one of the main ones, the most important one that we focus on is sodium. So again, we want to know, okay, where is sodium mainly found, inside or outside the cell, and how do we control it? So with sodium, there are different types of receptors that are sensitive to, for instance, the concentration of solutes in the fluids. There's also this RAS mechanism, you'll remember, right? RAS is renin, angiotensin aldosterone system. And so it's initiated by the kidneys, and ultimately it actually has an effect on the kidneys that then works to modify the amount of sodium that is reabsorbed. And because it's a sodium-potassium pump or an exchanger that controls that, that also influences the amount of potassium that is secreted. So we see here that aldosterone is the major hormone that is exerting this effect on the kidneys to allow us to reabsorb more sodium. So sodium is an important one, like we said. Aldosterone helps to control it. We also find that there are other hormones besides aldosterone that we'd heard of. This one, atrial natriuretic peptide, ANP. And we see Na, right? Natrium refers to sodium. And so ANP actually has an antagonistic effect to the RAS mechanism, or aldosterone. So what we found before is that we were going to be holding on to more sodium, holding on to more water, and increasing blood volume, etc., and blood pressure. In this case, we have the opposite effect. We want to get rid of some excess instead. So you should know those two hormones and their effects that they have. We also look a little bit in terms of potassium. Now, potassium is going to relate to sodium levels, but see these bold terms here, you should know these as well. Remember, hyper is going to mean a lot. Hypo means less or little. So kalemia refers to potassium levels as opposed to natremia for sodium. Calcium is our third important ion that we regulate. And so again, we need to know what it's used for in the body, and how we control the concentration of it. And is it found in the extracellular or the intracellular fluid, which you can see a bit right there. And this is the way that calcium is controlled, mainly by parathyroid hormone from the parathyroid gland, as well as, in extreme cases, calcitonin is going to contribute as well from the thyroid gland. Now this psychedelic picture here transitions us into the next segment where we're also talking about electrolytes, but in this case we're talking about things like hydrogen ions, right? So if we have an electrolyte, a charged particle that when you put it in water it breaks apart into ions, if in fact one of those ions is a hydrogen ion, then that is going to affect pH. Oh, you can almost see that still on the screen there, huh? Okay, so you should know the pH scale. You should know the normal pH of bodily fluids, particularly of the blood, because this is very, very tightly controlled. So you can see it centers around 7.4 on the pH scale, so it's slightly basic. But you can see it's very tightly controlled. So if it goes a little bit above that or a little bit below that, we end up going into acidosis or alkalosis. And so if that happens, we have to try to nudge the body back into homeostatic control, get it back towards the set point. And we have three major mechanisms to do so. Some are faster than others, right? Like the chemical buffer systems. Other ones are slower, but they may be more effective. And the mechanisms that we see in terms of respiratory system and renal, right, urinary system, we've been introduced to those before. So these chemical buffer systems are really about the only new information that we have in terms of controlling hydrogen ion regulation. So we do some review between what makes a strong and a weak acid when these dissociate into free-floating hydrogen ions like you see here. And then we just describe examples of these three lines of defense in terms of maintaining pH in the body. So here's some of our examples in terms of things that can either bind or free up hydrogen ions. So the bicarbonate buffer system, this is one that we talked about with the respiratory system, the phosphate buffer system, and the protein buffer system. All three are things that can bind up or release hydrogen ions as necessary. And this equation, right, this is the bicarbonate buffer system that we see here. This equation is very important. So it's what's occurring in the red blood cells when we're changing carbon dioxide into bicarbonate ion. It's what's happening associated with the respiratory system in terms of transporting carbon dioxide around the body. So it's used in a number of different instances. So that counts as both a chemical buffer system as well as part of how the respiratory system contributes as well in terms of controlling acid-base balance. And then last is going to be the renal mechanisms of pH balance. So you need to know those three, be able to describe how each of those three contribute to a balance in pH. And then be able to give a definition for, like we said, respiratory acidosis and alkalosis. And then what we'll find is metabolic acidosis and alkalosis. Effectively, if one of these three systems, right, like we have the chemical buffer system, the urinary or the renal control system, and then the respiratory control, if one of those goes down, like the lungs are not functioning properly or the kidneys are not functioning properly, then the other two out of the three work to try to compensate. That's the gist of what we find here. Okay, so there is your tour through this, like we said, synthesis chapter where we're looking at the control of fluids in the body and solutes, including pH. So I hope that helps out, and we'll talk to you again soon.